Assalamualaikum and hi, my name is Iman Nur Aina, and in this video, I am going to present about my final year project titled Comparative Study on the Chemical Profile of Stainless Steel Propolis Extract Trigona apicalis from different locations. As for introduction, propolis is a resinous substance produced by bees by combining plant resins with their saliva and enzyme secretions. Its composition, which includes bioactive compounds such as flavonoids, terpenes, and polyphenols, varies depending on the plant's and geographical region. Propolis is widely recognized for its antibacterial, antioxidant, and medicinal properties. Despite its potential, there are significant gaps exist in Malaysia, particularly for Trigona apicalis propolis. First, there is no established quality control standard for Malaysian stainless steel propolis, as it is important to ensure the quality, efficacy, and safety of propolis goods. Second, comprehensive chemical profiling for this species is lacking, thus causing a research gap that will hinder our understanding of pharmacological and therapeutic properties, as the chemical composition has a strong correlation with pharmacological properties. Lastly, comparative studies that investigate how the chemical composition of propolis varies across different locations is one of the problems arise, cause it will hinder a study in defining which locations produce high quality propolis and can be further used for researchers and producers. To address this issue, this study was conducted in order to determine the chemical profile of Trigona apicalis propolis from selected locations around Jengke Pahang using analytical techniques such as thin layer chromatography, Fourier transform infrared spectroscopy, and ultraviolet visible spectroscopy. The second objective was to identify correlations between the chemical composition and geographical origin through chemometric techniques, including principal component analysis and hierarchical cluster analysis. First, in order to determine the chemical profile of Trigona apicalis from selected location around Jengke Pahang, thin layer chromatography was used to separate compounds on silica gel plates under UV light. As we can see, the developed TLC plates observed under 254 nanometer UV light show several black spots. These spots indicate the presence of compounds with aromatic rings or conjugated double bond. For 366 nanometer UV, there are several spots visible exhibit fluorescence. Fluorescence is the emission of light by the substance that absorbs UV or radiation. Phenolics and flavonoids that are commonly found in propolis are among the substances that are able to exhibit fluorescence. The developed TLC plates were further observed with anisaldehyde and aluminum chloride derivatization. The purple and pink color appeared and the developed TLC plate was sprayed with anisaldehyde. Reagent indicates the presence of terpenoids and phenolics. The presence of flavonoids from yellow, blue or yellow-green spots observed under long wave ultraviolet light after being developed in aluminum chloride reagent. According to Badi Azaman 2023, purple spots appear after anisaldehyde derivatization indicate the presence of terpenoids. From the TLC chromatogram, it is safe to say that terpenoids are the major components in Trigona apicalis propolis, aligned with the major purple spots visible. According to Ristivojevic 2017, UV spectroscopy is highly suitable for the investigation of phenolics such as flavonoids due to the presence of conjugated aromatic rings substituted with OH groups. The UV spectral profile of location 2 and location 3 should to be similar, suggesting a homogeneous chemical composition among the samples with maximum absorption at 340 and 336 nanometer. Location 1 propolis had lower maximum absorption at 281 and 268 nanometer. Bazauhin 2021 stated that 4 samples of eternally eastern Algerian propolis obtained 2 absorbent peaks at band 2 between 200 and 250 nanometer and band 1 between 300 and 350 nanometer, which correspond to the flavonoid family. For FTIR analysis, wave number of 3550 until 3200 was assigned to OH stretching vibrations and may be seen in all locations represent hydroxyl groups that one of the main compounds in flavonoids and phenolic. The C double bond O stretching indicates carbonyl group can be seen in all locations. Phenolics compound in propolis can be determined at location 1 and location 3, which correspond to CO stretching of phenol group. However, location 2 propolis has no peak around those values, made due to an absence of homogeneous phenolic compound as is in location 1 and location 3 propolis. There are several minor peaks with weight absorbance around 1,300 and 1,000 that can be observed in all locations, correspond to terpenes compound in those propolis. In order to identify the correlation between chemical composition and origin of propolis, chemometric analysis has been conducted by using PCA and HCA. PCA or FTIR spectra showed the scattering of propolis samples along the principal component 1 and 2 based on functional group with variance at 100%, which first and second species displayed variability of 58.67% and 41.33% respectively. The samples from location 1 and location 3 clustered closely together in PC1 correspond to the homogeneous of phenolic compound determined at these peaks. Location 2, however, was positioned separately along PC2, suggesting a distinct chemical profile influenced by the plant source unique to that location. As for HCA, the dendrogram shows the propolis extract which were clustered into two main groups based on the HCA plot. Location 2 and location 3 propolis shared more similarities in their chemical profiles due to the influence of common plant source as they were classified under the same cluster. This was aligned with reported study from Badi Azaman 2023, which the grouping of Trigona apicalis in HCA are based on the plant source available in each location, as well as intensify of their FTIR signals. The similarity between location 2 and 3 suggests a shared vegetative influence, while location 1 stands out due to its unique chemical profile. In a nutshell, our research successfully identified the presence of vital bioactive compounds such as terpenoids, flavonoids, and phenolic compounds in propolis samples from selected locations around Jengkel Pahang. These compounds were confirmed through advanced techniques like TLC, UV-Vis, FTIR, and even chemometric analysis. Hence, contribute to providing valuable insights into the standardization and quality control of propolis for its potential application in cosmetics and healthcare industries. Looking ahead, chemical analysis by the integration of gas chromatography mass spectrometry or GCMS and machine learning to further enhance the understanding of propolis composition and its pharmacological benefits. With continued research, stainless steel propolis holds great promise as a sustainable multifunctional bioactive resources.